Welcome back, folks, to part two of our notes on natural selection. In this part, we'll be focusing on the Hardy Weinberg equation, population genetics, and variations in populations. I'm going to do things a little out of order for how AP Bio puts it. Order I think is a little bit makes a little bit more sense to me anyway. So we're going to actually start with the Hardy Weinberg 7.5, then we'll do 7.4, and then 7.12, 7.12. Even though that's the last one, but I think they kind of fit better here together. So that is what we're going to be doing here. So starting out with Hardy Weinberg and population genetics, you can see the parts of the book that go along with that, 23.2, 23.3, and 23.4. All right, so first thing, you've probably heard of the term of a gene pool, and I think we've talked about allele frequencies. Yeah, we did talk about allele frequencies in the last set of notes. So a population is a localized group of individuals capable of interbreeding and producing fertile offspring. So you got to be able to be close enough with somebody to regularly mate within that group, and that your offspring should also be able to mate within that group. That would be the definition of a population. Every population has a gene pool, okay? And a gene pool consists of all the alleles for all loci in the population. Keep it simple. It's all the alleles for all the genes in the population. Each gene is a loci. That's a little spot on the DNA where that gene resides, and you've generally got two alleles for that gene, right? So that's the loci, is where that gene is on the DNA, and you have two alleles for that gene. Now that might be the same alleles, homozygous, might be heterozygous, whatever, like we learned about before. So that's kind of what we're trying to show down here is the gene pool, the different alleles that are available in a population. Here we can just see we've got a little bit of genetics going on here. We've got red flowers, two big R's, white flowers, two big W's, and pink, a red and a white mixed together. So, some terminology. A locus is said to be fixed if all the individuals in a population are homozygous for the same allele. That means there's no difference. Every single person for both their alleles has exactly the same DNA. Okay, there's no dominant recessive or anything else. They're all big C, big R. Nothing else. That's it. That's fixed. There's no room for evolution to occur on that particular gene and allele, that locus, because there's no variation on that gene or locus. And that can happen in populations. If there are two or more alleles for locus, and we're going to stick to talking about just two, just to keep things easy. Diploid individuals may be either homozygous or heterozygous. They could be big R, big W, or big R, big W, right? So that's kind of the more common situation that we'll be discussing here in this unit. We can calculate the frequency of an allele in a population. Frequency is just like essentially what percentage of the alleles in a gene pool are the big R and what percentages are the big W, okay? So the total number of alleles at a locus is the total number of individuals. Say if I got 10 individuals in a population, there would be each individual has two alleles for that gene. Okay? So you take 10 times 2, and that gives you the fact that there's 20 different alleles to choose from in that population. Okay? The total number of dominant alleles at a locus or for a gene is 2 alleles for each homozygous dominant individual. Okay? So if I was the two big R's, right? Or homozygous recessive, two little R's, and one allele each for heterozygous individual. They'll have a big R and a little r, kind of a deal. By convention, if there are two alleles at a locus, we'll call them P and Q to represent their frequencies. P is generally the, if there's a dominant recessive type situation, P is generally the dominant and Q is the recessive. I'm not sure that you really didn't know that, but that's kind of the general convention there. So if you do that, as with any frequency, if you total up the frequencies of P and the frequency of Q, it should equal 1. Like if we're 60% big R, we should be 40% little r, so we add up to 100% of the alleles in the population. So we're going to work through an example, and this is the one that's in the book, so you Did this on purpose so you can go back to the book and kind of compare back and forth and read what they've got, and it's all in the same language here for this example. So let's say we've got population of flowers that looks like this. These are the genetics available in the flower. We got 500 total flowers in the area, which means we have a total of 1,000 alleles, because each of these flowers has 2 alleles for that gene, that color gene. All right, so let's say we have 320 red flowers, so this would be their genotype. We have 160 of the pink ones down here, that's their genotype, and 20 of the white flowers with this genotype. That's just the distribution we happen to have in this population. If we want to calculate the number of copies of each allele, how do we do that? Well, for this allele, the big C, big R, we know we've got 320 who have 2 of those. So 320 times 2 gives us how many C, big R alleles we have in this group. Plus, in the heterozygous group here, we've got 1 of those in every individual. So that's 160 of those. So we add in those 160, and we get 800. Okay. For the CWs, we got 20 of the ones that have 2. So 20 times 2 alleles in each one of those individuals. 160 that have 1 of those Ws. So we add that in there, and that's 200. So our frequencies, we got 800 and 200, and you can probably do the math. This would be 0.8 or 0.2. Okay, so frequency is the number of occurrences of the thing you're interested in divided by the total number of occurrences. So it's just like figuring out your percentage on a test. What percentage did you get right? I got 8 out of 10 right. 8 of the thing I'm interested in out of 10 total possibilities. Same thing here. We have 800 of that allele that we figured out, divided by the total number of alleles, which is the 800 in the R and the 200 of the W, or 1,000. So 800 divided by 1,000, 0.8. Same thing here, 200 divided by 1,000, 0.2. If you add those 2 up, we should get to 1. So it all checks out. It's internally consistent. All right, and that brings us to the Hardy Weinberg equation. Okay. The Hardy Weinberg equation describes the genetic makeup we would expect for a population that is not evolving at a particular locus. In other words, a population where the allele frequencies are staying constant. This generation is 80% red, the next generation is 80% red, 30 generations from now it's 80% red. Okay, no change in allele frequencies. If you don't have a change in allele frequencies, you don't have evolution, because that's what evolution is. Remember, that's that microevolution we talked about before. So this is describing a situation where there's no change. Okay, so I kind of like think of the Hardy Weinberg as a null hypothesis. This, if there's nothing influencing the evolution and no evolution is happening, this is what we'd expect. So it's kind of like a null hypothesis in that respect. So bullet number 2, if the observed genetic makeup of the population differs from these expectations, it would suggest the population may be evolving. So if we do this equation and it's different than what we'd expect, what we see is different than what we expect according to this equation, then it might point to, hey, there's evolution going on at this locus, for this gene. Now, begs the question for you guys, how would we what test would we use to see if that difference that we're seeing is significant difference? You could use the chi-squared test, right, to see if it's a significant difference. Because, I mean, this is like anything with statistics, guys. We can see that there's a difference from expected, but then the next step is it a significant difference from what's expected? Then you would apply the chi-squared test if you're trying to go that deep into the problem, which I don't think we're going to in AP Bio. Don't think they're ever going to ask you to do both of those on one problem. All right, so let's kind of talk about the Hardy Weinberg equation. So in order for this to happen, it has to be where the gametes contribute to the next generation randomly. So there's no like, oh, I pick who I want to mate with based on the way they look or anything else. It's just 100% random mating, random gametes. Like, you take all the eggs, and you take all the sperm, and you throw them in a bucket, and you let them mix. You get something similar to this with the way fish, like salmon, breed. All the females lay all their eggs, and the males come in and squirt their sperm all over the top, and there can be some of that randomness there. Okay, And then, of course, we got to have Mendelian inheritance happening. If that's the case, allele and genotype frequencies should remain constant from generation to generation, hence no evolution. So here is the equation. This is what is on your, this is the reason it's such a bad view, is this is literally taken from the PDF of your AP cheat sheet. Here's the equation. P squared plus 2PQ plus Q squared equals 1. And then the reminder that the frequencies should always add up to 1, so P plus Q equals 1. P, here and here, is the frequency of allele 1 in a population. So in the one we were looking at before, it was big R. And Q is the frequency of allele 2 in the population. So what we were looking at, that would have been the W allele. Now let's start working through this a little bit. So like I said, it describes a constant frequency of alleles in a gene pool. No change. Over here, we can see here's that pool, right? 80% of these are red ones, 20% are white ones. So any given time, you're going to mix things. You got an 80% chance of an egg coming out, and you got an 80% chance of a red sperm being the one that's given, etc. So same example from before, 500 wildflowers, 1,000 alleles. Our CR was 0.8, and our CW frequency was 0.2. We are doing our P to Q already. So we can calculate the frequency of the genotypes. All right. So what frequency of the genotype should be big CR? Okay. If the allele for just one CR is 0.8, what are the chances we're going to get a CR in the egg and then a CR from the sperm? Well, that's the multiplication rule from genetics, right? The chances of this and this both happening, we multiply the chances of each one singularly. So that would be 0.8 times 0.8 equals 0.64. The Hardy Weinberg equation just simplifies that to P squared. The frequency of P allele, which is C big R, and we square it because that's this right down here. So P squared, 0.8 squared is 0.64. Same thing down here. I'm going to skip all the way down to the W's. Okay, the chances of being homozygous W is Q squared, 0.2 squared, 0.04. And the one in the middle here, the heterozygous situation is, okay, what's the chances we're going to have a CR and then the sperm comes in and it's going to have a CW? Well, that's 0.8 times 0.2. But we also have to add in, what are the chances it went the other direction? What if it was a CW in the egg and then a CR in the sperm? We do the same thing. We just flip the numbers, right? But we still wind up with 0.8 times 0.2. So we can simplify that to, here's our 0.8 times our 0.2. Then we multiply it by 2. One for this and one for that. So if we do that, we get 0.32. And if we add those all together, we should get 1. And if you want to take the time, you can take a look over here and you can see how that matches up with what we see in a Punnett square. So you can kind of prove this equation using a Punnett square over here. So if P and Q represent the relative frequencies of the only two possible alleles in a population, a particular locus, then that's our equation. That's the frequency of one allele, that's the frequency of another, and then you square it, you get all this, and it should add up to 1. If things are constant. So P squared and Q squared represent the frequency of the homozygous, right? Because this was RR, this was WW. 2PQ represents the frequency of the heterozygous genotype. 2 times the frequency of R times the frequency of W. Oh, there, had that there. So here's another way to think of this, guys, because it can be helpful, because sometimes I'll ask you what the frequency of the homozygous is. And that you would take if you knew the frequency of P, you just square it and you can get that. The frequency of this genotype is here, and the frequency of the homozygous recessive phenotype in this case, or the second allele in this case, is Q squared. So this allows us to do some basic algebra to kind of figure out any of these different variables depending on what we know. Okay. So let's take a break from the math for a second and come back to the theory. So the Hardy Weinberg theorem describes a hypothetical population that is not evolving. That doesn't really happen too much in nature. In real populations, allele and genotype frequencies do change over time. You may not see much of anything in a generation to generation, and not enough to detect it statistically, but they do change over time. So that begs the question, why would we bother with it? Because it's a null hypothesis, right? We can use it as our control to see if evolution is happening at a locus, at a gene, and or how fast it's happening, how quickly the rate of change is happening from the point of no change, the zero. So kind of like a null hypothesis. So there are 5 conditions that would give you a non-evolving population. Those conditions are rarely met in nature. Pause the video for a second and try to think of what conditions would lead to less to no evolution happening in a situation. All right, so we've unpaused it obviously here now. So here's what we've got. Okay, 1, no mutations. Okay, if you add in mutations, you get new alleles, and that throws the whole thing off, and you're going to get change. So in order for Hardy Weinberg to exist, that hypothetical no evolution, you got to have no new mutations. That doesn't really happen. 2, you have to have 100% random mating. There is no selection whatsoever. There's no behavioral selection, no sexual selection, no choosing a mate on any basis other than it's a warm body. Okay. The environment's got to play no role at all on who survives and who mates. So no natural selection is happening. Anybody who lives or dies is complete random chance. This is generally only possible in extremely large populations, because as we'll learn later, really small populations, there's some other weird things that can happen statistically. So we got to be looking at a pretty large, stable population that doesn't have any gene flow, meaning you're not getting new genes in from outside the population that are going to add new genes, because we don't want to add new genes. We don't want new mutations. New genes are like new mutations. We can't have outside DNA coming in. And these things all happening, very rare. Now, it is possible for natural populations to evolve at some loci, some genes, but not at others. So they're being Hardy Weinberg equilibrium at some loci, but the chances of them being at Hardy Weinberg in all loci is pretty much 0. All right, so let's apply this to a genetic disease, PKU, phenylketonuria. Okay, my brother-in-law has this. This is a genetic disorder in which the individual is not able to correctly process in the digestive system phenylalanine, that one amino acid. And then that one amino acid gets processed into a neurotoxin, and it winds up being degenerative neurological disorder. If you know the person has PKU early enough, you can provide them the correct diet that has all the amino acids but phenylalanine, and then they're fine. They got to be on a very strict diet, well, especially while they're developing. Once they're fully grown, it's not as big a deal, but while they're developing, it's a pretty big deal. So if they're on that diet, there's no real drawback. They'll survive, they'll be able to reproduce, all that kind of stuff. Okay. In the past, they'd die pretty young. This was one of the first blood tests that they did on babies right after they were born back in the 60s to develop it. And they'd take a little blood from the baby and they'd test it before they let the mother feed the baby. Because the mother started feeding the baby breast milk, it would have phenylalanine in it, and that baby would start to get brain damage. So the mother would essentially be poisoning the baby. They'd have to get the baby on that. So my brother-in-law is a bit older than me, and he was, if he was too much older than me, he would have been before this and he wouldn't be around. But since this test existed, they caught it. He had a very strict diet when he was younger, so he is still around right now. So this PKU allele seems to be in Hardy Weinberg equilibrium because the PKU gene mutation rate is very low to non-existent. Mate selection is generally random with respect to whether or not an individual is a carrier for PKU, because you got no clue somebody's a carrier. It doesn't affect them in their day-to-day life. Okay, and then it doesn't really, and if they've got a good diet when they're younger, it doesn't really affect them, and you don't notice it. Natural selection can only act on a rare homozygous individual, and then only if you don't catch it and they are eating a bad diet, which in the United States we generally catch. The population in the United States is very large, and migration has no effect, as many of the other populations that are moving in and out of the United States also have similar allele frequencies for PKU. So it's not the in-migration, out-migration doesn't affect the gene frequencies. So let's take a look at this. Let's use these equations in this situation. Okay, so the occurrence of PKU is 1 per 10,000 births. Okay, 1 per 10,000 births. So PKU we're looking at here, we're looking at the recessive trait in this case, so we're going to use Q, not P. So the frequency of homozygous recessive, which is what you have to be to get PKU, is 1 in 10,000 births, or 0.0001. That is the frequency of that genotype. Okay. So if we want to figure out the frequency of Q, we need to take the square root of this, right? Because Q squared is equal to that. So the square root of this is Q. So the square root of 1 in 10,000 is 0.01. So now we know what Q is. If we know what Q is, we can figure out P pretty easy. Okay, so the frequency of the normal allele is 1 minus Q, right? Because it should always add up to 1. So 1 minus Q, 1 minus 0.01. So the normal allele is in 99% of the population. Now that we know the frequency of both P and Q, we can figure out the frequency of carriers. This is the carrier genotype, right? It's 2PQ. So it's 2 times 0.99 times 0.01, which is the Q, right? So the frequency of carriers in the population is around 2%. Okay, so 98% or so of folks, a little less, aren't even carriers for it, don't have the allele at all. 2% are carriers, and less than 1 in 10,000 actually has PKU as a result. All right, now you should, we're done with the Hardy Weinberg stuff, guys. Go ahead and we've got some things here. So on page 487 in the book, we've got this checkpoint question, which I'd like you to do. So write that number down. We have question 2, this question. And then question 3, this question. If you're in class, we will go over these together once you've done them. If you're not in class, I will post a separate thing that talks about the answers on this. So we'll check you later, guys. Have a good one. The other thing you should do, don't worry about that. Just a reminder on what null and alternative hypotheses are. And then you should also do topic questions 7.5, because that's the stuff on the Hardy Weinberg. So do topic question 7.5. And that will do it for this chunk of the notes, guys. Meet me back here in a little bit, and we will go through how to alter allele frequency in the population. So we'll be going through the 7.4 and 7.12 stuff. Okay? So I'll see you then. Bye.